Students, today our class deal with epistasis, or we can call it as epistatic gene interaction. Then what is epistasis? Epistasis means one gene can, here it is a non-allelic gene interaction. Here one gene can suppress the effect of another gene. So what is the term? Suppressed. One gene can suppress the effect of another gene. So, if there is two genes, capital A and capital B is there, which are two non-allelic genes, and if capital A is present, what happens is that even though the capital B is present, the phenotype of capital B cannot be expressed. Phenotype of capital B cannot be expressed. That is epistasis. Here, one gene can suppress the effect of another gene. For citing that, I am telling, even if there is two genes, capital A and capital B, capital A can have the power to suppress the expression of capital B. So, if capital B has a particular phenotype, but that phenotype cannot be expressed due to the presence of this gene. So, one gene suppress the effect of another gene. So, that is here we can termed as one gene is epistatic gene, and another gene is hypostatic. How we can say that? Which is the epistatic gene? Capital A is the epistatic gene, and which is the hypostatic gene? B is the hypostatic gene. So once again, what is epistasis? Epistasis means expression of one gene is suppressed by the expression of another gene. And students, we have to study two types of epistasis, that is, one is dominant epistasis and another one is recessive epistasis. In dominant epistasis, the dominant gene controlled the expression of another gene. Dominant gene can control the expression of another gene. That is, if a dominant gene is there, then if one of the dominant gene is there, then whatever may be the second gene, the phenotype may be of same. But in the case of recessive epistasis, recessive gene is there. So homozygous, if the homozygous recessive gene is there, then what happens? If whatever may be here, if it is capital A or if it is small a, or we can say as whatever may be it, the first gene, the expression will be of same. So, in dominant epistasis, dominant gene can control the phenotype, and in recessive epistasis, homozygous recessive gene can control the expression. So, then what is the phenotypic expression of dominant epistasis? That is 12:3:1. What is the recessive epistatic phenotype? That is the 9:3:4. Then here the 9:3:1 is the Mendelian dihybrid ratio, and here the 9 and 3 phenotype, that is the two different phenotypes combined together to form a single phenotype, and we get 12. So 12:3:1. In case of recessive epistasis, the last double homozygous recessive one and 3 of the another phenotype combined together to give one phenotypic expression, that is the 9:3:4. So students, in dominant epistasis the ratio is 12:3:1, and in recessive epistasis the ratio is 9:3:4. Okay. The example for dominant epistasis. The example for dominant epistasis, we have to study summer squash, that is Cucurbita pepo is its scientific name. And here when we observe the summer squash, there is three different morphology of fruit color is noticed. Three different morphology are white, yellow and green. And this white, yellow and green are the three different phenotypic morphology of Cucurbita pepo, or which we call it as summer squash. And here Bateson conducted the experiment, and what he find out is that he selected two different morphologically different varieties of Cucurbita pepo, as one is white and another is yellow. For getting ratio 16 progeny, we have to get a heterozygous F1. For getting heterozygous F1, what we have to do? We have to select the plant of this white and yellow as both gene heterozygous case. Here there is two, we can write the three types, two genes are there. They are W and Y. W and Y. Why? If W is expressed, white is expressed when there is capital W is there, and if small y, and we can write it as small w, capital Y, capital Y. Why we write this expression, genotypic expression? Because even if capital W is there, what is the phenotypic expression? Since it is an epistasis, the phenotypic expression will be white. So we write it as small w, capital Y, capital Y. So here we get capital W, small y as the gamete, and here we get small w, capital Y as the gamete. When they fuse, we get capital W, small w, capital Y, small y. This is the F1. This is the P1, parent. This is the what is the phenotype of this F1? Even though capital W is there, since capital W is there, even if capital Y is there, the Y has no effect. Only capital W has effect, because capital W can suppress the expression of capital Y. So the expression is white. So expression is white. Here we get heterozygous progeny. Since it is heterozygous, when we take this type of another type of this white, how many gametes will we get? Since it is heterozygous heterotype, we get 4 different gametes will be produced. That is with the capital W, capital Y, capital W, small y, small w, capital Y, small w, small y. These are the 4 different gametes produced from this white F1 heterozygous progeny. Okay. Okay students, for easiness I have written it like that. The gametes, there are how many different gametes? There are 4 different gametes. So we can write it as this. And when we write the progeny genotype, how we can identify the different phenotype to fit that ratio. So I have previously said that even if one gene, that is W, is in the dominant state, then what will be the phenotype? White. If W is absent or capital W is not there, the expression of Y gene will be there. So in here we have simply understand that here there is no capital W gene, And again here there is no small capital W, only small w, again small w. Then what is the another gene? Capital Y is there for here, here and here. So these three progeny will be yellow colored with yellow colored fruit. Yellow colored fruit. But here, since yellow color is absent, why? Capital Y is not there and there is no capital W. So the fruit color will be green. Green in color. Again, then what is all these? There are all cases. What is there? There is capital W gene. At least one capital W is there. Here it is. Here it is. Here it is. Here it is. So these all 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. So 12 is white is to 3 yellow is to 1 green. So the condition is that if 12 means 12 capital W dash 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 dash, we never bother whatever it may be, is to 3 small w must be essential. Then one of the capital Y gene is also essential. So another gene may be either capital Y or small y to get yellow phenotype. And the last condition is 1 is small w, small y. This is the condition to get the expression. So students, there will be at least one capital W gene is there to express Y. So here all are, we can write white, 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 white. You also write it in the book and understand how these genes get interacted. White. Don't be by-hearted and write this. You have to simply identify where is the difference. Here the difference is here, these 4 columns. So this is the dominant epistasis. Okay, today I am winding up. So what is the phenotypic ratio of 12 is to 3 is to 1. Thank you students. Hope you are all safe at home. Okay, bye.